literature review over the summer with my mentor, Dr. Amrita Sewell, a postdoctorate student from Yale University, and the literature review was focused on Li-Fraumeni syndrome, cancer, and how it can be paired with gene therapy for possible treatment. So I chose this topic because I was interested on how cancer could be treated using gene editing, and my mentor suggested I focused on brain cancer within Li-Fraumeni syndrome. So here's a quick overview of the research that I learned from this. So cancer is a disease that affects the human body by causing malicious cells to grow and spread uncontrollably, affecting the body in various ways. If these cells lump together to form lumps of cancerous tissues or tumors, the consequences could be life-threatening. With over millions of individuals being diagnosed with cancer a year and over 600,000 dying, it is important to find a treatment for it. Now, scientists are looking towards gene therapy for a possible treatment, as genes play a crucial role in individuals developing cancer. So one type of brain cancer is obviously says that is brain cancer, which is considered to be one of the deadliest types of cancers. It is extremely difficult to treat due to the natural blood-brain barrier, a complex web of blood vessels, which helps keep the brain safe by keeping any harmful substances away from the brain and only absorbing nutrients. Unfortunately, this will prevent any type of anti-tumor or anti-cancer drugs from reaching the tumor, which is behind the barrier, because the blood-brain barrier identifies the drugs as unfriendly and will not let it pass. Additionally, it is very difficult to remove the tumor in a craniotomy, a surgery that concerns opening the skull, as the brain is more sensitive compared to other organs and cannot have the surrounding tissues around the tumor be removed, because this is a method used to ensure all cancer cells are removed from the infected organ. Fortunately, though, when a surgeon removes most, if not all, of a tumor, the amount of pressure of the brain is lifted, and the amount of tumor needed to be targeted is lessened a bit. So, Li-Fraumeni syndrome is a rare inherited disorder that causes mutations in gene TP53. This will increase risk in half of all types of cancers with 140 plus types of mutations. Gene TP53 is a very important gene because it instructs the creation of protein P53, which is a tumor suppressing protein. P53 is nicknamed the guardian of the genome and helps destroy damaged cells, preventing tumorigenesis. It destroys damaged cells by instructing apoptosis, or basically the self-destruction of a cell, and will effectively stop the mutated cell from dividing and passing on its damaged DNA. Without P53, abnormal cells will divide without any type of restriction and possibly become cancerous. Currently, most treatments, quote unquote treatments, because they're not really treatments, will focus on prevention, such as annual screenings with doctors and leading a healthy lifestyle. However, in recent years, gene therapy has become more and more popular in the medical field as it opens up many different possibilities and treatments for various conditions. There are currently two types of gene therapy, somatic gene therapy and germline gene therapy, and each way has a different way of approaching treatment. Somatic gene therapy is when a section of DNA is put in a cell where no sperm or eggs are produced. Thus, the effects of the gene therapy on the patient will not be inherited by any children. On the other hand, germline gene therapy has DNA placed in the cell where sperm or eggs are produced, and these changes will be passed down to the individual children. To carry out this, gene augmentation, gene inhibition, or killing of certain cells will be needed to be used. This is when a malfunctioning protein is replaced, suppressed, or destroyed, respectively in that order. Unfortunately, this technique is still pretty underdeveloped, and there are still obstacles that need to be surpassed, such as ensuring the cell will not reject the cell-containing therapy. So, after researching this topic, my mentor and I had a section where we kind of discussed the next steps in creating a gene therapy treatment for Li-Fraumeni syndrome or cancer in general. We believe that gene augmentation or inhibition is the most possible route for this, as almost 2 trillion cells divide in the body per day, making killing of all the cells extremely difficult. Through this, newly created cancer treating drugs can also possibly make their way through the brain because it can be disguised to be a cell from the body and can trick the blood-brain barrier. So my experience with Talaria was actually really beneficial. Being able to work with someone who had years of experience and plenty of knowledge in a field that I was really interested and relatively new to helped me go deeper into the field. I came out of the program knowing a lot more than I did when I went in, and the information was all really helpful and interesting. My mentor was really patient and helpful throughout the whole process and sent me different resources and helped edit and give me suggestions for my paper. I'm really grateful for everything she has done for me throughout the 6 years. I was also able to gain, as I said before, a lot more knowledge in this area through the program. Previously, I had only known really basic information about cancer, and I didn't even know about Li-Fraumeni syndrome. Well, I did have some difficulty understanding different types of genes because I had to go through a process where to pick a gene to focus on for the literature review. I had some trouble figuring out what meant what and like how they were named, but she was really helpful and I was able to understand through different resources and databases. So thank you, and Talaria was a really good step in my future research and a good way to experience working with her.